everyone. Today I'm going to talk about the sickle cell disease, also known as sickle cell anemia. And there's a lot of interesting things that we can consider and think about as researchers, as genetic counselors, as some of you are or will be in the future. So I have a couple questions at the end of my presentation to engage some kind of discussion here. But basically, it is a genetic disease in which the red blood cells take on a different shape, more like a sickle. So given the importance of our red blood cells, the fact that this new shape doesn't do its usual job very efficiently creates a lot of problems. But basically, it is caused by a defective hemoglobin, this cute protein right here. And actually, it is made up of 4 different polypeptide chains that make up the quaternary structure of this protein. So just a quick reminder, not all proteins adopt this quaternary structure. Some of them stop at the tertiary structure, some of them stop at the secondary, some transmembrane proteins. But anyways, these four proteins are the alpha and beta, and they are connected by an heme group with iron atom in the middle, which gives the taste of iron when we taste blood, when we bite our tongue, for example. But different hemoglobins have different combination of these chains, which is interesting when we think about other hemoglobin disorders, because there's more than sickle cell disease, right? Like thalassemia, if I'm not wrong. But the fact is, we have a lot of other hemoglobins, but the one that is important here for sickle cell disease and disorder, it is the hemoglobin A, also known as HbA, which is made up of 2 alpha globins and 2 beta globin chains. And what happens here is that the beta globins are misshapen for a very small, or maybe not that small, detail, right? The gene that codes for this beta globin, the HBB gene, has a mutation on the second base of the code, which changes completely the amino acid at the translation process. But this gene is located in the chromosome 11, which makes it an autosomal trait. And to develop a sickle cell disease, you need to have both chromosomes with the mutated gene. So it is a recessive disease. So you need to have these 2 copies, and which makes you a homozygous for this trait. And for every homozygote, we have a heterozygote as well. So in this case, they are carriers of the gene, carriers of the trait, but they don't express the sickle cell trait. But that doesn't mean that they are free of effects, right? Especially if they are in some extreme conditions. For example, high altitude, which makes sense considering that only one gene produces healthy hemoglobins and the other one produces the sickle ones, which would confer the person less oxygen to their cells, right? Because they're not that efficient. And actually, it can actually carry the oxygen, but the problem is when the oxygen leaves the protein, which makes the hemoglobins to kind of attach one another and form some other structures, which can actually block the blood flow. No? But the mutation that causes all this is a non-conservative missense, which changes one amino acid to another. Remember that I told you that it changes one nucleotide that codes for the amino acid? Yes. So the glutamic acid is replaced by the valine. So here we have an adenine being substituted by the thymine. And if we look to these 2 amino acids, we can see that the valine has a hydrophobic group, instead of hydrophilic group as the glutamic acid. And this changes everything, right? Considering that now this part of the protein will tend to hide from the water molecules because it's hydrophobic, and it changes completely the conformational structure. And actually, what fascinates me the most about molecular biology is how much a single detail in our eyes, it is a single detail, just a comma, but this can change everything, right? Proteins can have altered binding sites, lose activity, play different roles in a cell. So it's fascinating. But the fact is, this affected hemoglobin A is called HBS, and it causes tons of cascading events and effects. But the one that worth mentioning is the vaso-occlusion, right? That causes the traditional pain crisis that these people fortunately have to experience, and other underlying effects. But carrying the sickle cell trait is advantageous in some regions where this silly protozoan here is present and causes malaria. So since it can be carried well by all hemoglobins, the person has a higher chance of surviving. And this is actually evolutionary response to a selective pressure, and this selective pressure is the malaria. So because it was beneficial, this gene that started, mutated at some point in the evolutionary history, and probably raised in Africa, some part in Africa, it increased the frequency because it kind of allowed people to survive in this malaria environment. And you might think, okay, so this is only a problem in these places in Africa, right? Wrong. Because even though this mutation happened, and actually it is present in Africa, some parts of Asia as well, unfortunately, with the sad historical moment of slavery, all the countries that received a huge amount of people from this region are facing this health problem today. So what I want to bring for discussion here is it a racial disease? Like, we know that it's more common in people from African descendants, but what else? Are our phenotypic differences a signal of what may or not be on our genome? Or, like, what I mean by this is, can a Caucasian phenotype person be diagnosed with sickle cell disease, or even being a carrier? So I want to know what you think about it, and I would be happy to discuss with you guys. Thank you.